Hi, welcome. You have any questions, please do feel free to ask away. Chidima, how are you? Chidima, how are you doing? Oh, good, I'm pleased. What are you covering now in lessons? I am good, apart from I am cold. It's turned really cold now. Hi, Callum. Okay, so we just finished neuronal communication on one side. Nice. Love neuronal communication. Maybe one of my favourite ones, actually. On the other side, we're doing plant responses. Hi, can I ask a quick question? Of course you can. Please do ask away. Cold here as well. Honestly, my knuckles are, it's the worst in this time of year. My knuckles get so dry that they always end up splitting, and I hate that. So I need to really make sure I moisturise when I get in to the house. And I'm not sure if there's a delay in your message coming through. So, but please do, if you have a question, ask away. You hate the weather, but next human topic is finishing animal response. Oh, nice. Hi, Nina. Guys, if you have a topic area you want me to cover, then please do ask away. If you've got anything you're not sure of, please ask away. And obviously, please do come join me on the other side. Do you think that biologically speaking, it's harder for the body to get the arm to grow or the knee to grow? What an interesting question. Biologically speaking, we are talking about mitotic division occurring and then the differentiation of those cells. Do I think it's harder for the body to get the arm to grow or the knee to grow? Well, the knee's obviously got your patella and your cartilage extra parts, whereas with the arm, there's no extra part that's there. Also, but you're saying specifically the knee? I don't know. What an interesting question to ask. Please, could you explain the immune response? Hi, Simon. Yes, I can do the immune response. I haven't done the immune response in a while, so let's do that one. Let's start with that one tonight. I'm going to be pondering this one, that question you just asked me. That is going to make me ponder. Which do I think is better? Right. Okay, the immune response. Most thing when it comes to students struggling with the immune response, it tends to be understanding the one, different cells, and two, knowing the process that goes on. So let me just quickly pull over some of my parts to the slide, and then we'll go from there. Okay. I don't want that one, go away. I don't want that one. Well, no, don't want that one. I want it to be here, and I also need to just put this to my YouTube photos so I know what I've done. I'll quickly save it. There we go. Right, okay. I love that when it comes up. AI, what is it we're going to do? Ask about immune response. That's so funny. Right, guys, thank you for joining me. Okay, so what we're going to do today, we're going to look at the immune response, and we are going to, let me just pop you guys out from there, and hopefully I'll still be able to see your questions if you ask on that channel. Okay, so if we're looking at the immune response then, and we're talking about whether we have got the humoral antibody mediated response or whether we have got the cell mediated response, a lot of students struggle with the difference between these two. So the key thing for you to know when we're doing this is whether or not you're talking about just your B lymphocytes or whether you're talking about the T lymphocytes as well, specifically T killer ones. Yes, you can watch my content even though you're doing AQA. There's a lot of overlap between OCR and AQA. The only difference really is that they have certain bits where they'll go, yes, we really want you to say this, or we want you to say that. So it's just making sure that you are double checking your specification. Where I can, I will make sure that I give you what you need for both of them, but it is important that you do that double checking as well. Okay, so what have we got with the B lymphocytes and the T lymphocytes? Well, our B lymphocytes, I'll start with the B lymphocyte, and you should know that your B lymphocyte is going to be maturing in your bone marrow. That's why it's called the B lymphocyte. The T lymphocyte, that is going to mature in your thymus. So whether it's a B or whether it's a T is actually referring to where it's going to do the maturation process. So you know that cells go through mitotic division. Cells will then migrate through the body to where they will mature. So your hematopoietic stem cells go through that mitotic division to then get you into the sorry, I just read a question there and I was like thinking that one through at the same time. Try that again. So the hematopoietic stem cells are the ones that are going to go through that division, and then they're going to differentiate into whether they are a monocyte or whether they are a neutrophil or whether they are a B lymphocyte or whether they are a T lymphocyte. So whether they're a lymphocyte, and then where they're going to mature the B cells or the T cells is whether it's in the bone marrow or whether it's in the thymus. So one of the things to note is when we talk about our T lymphocytes and we're talking about the humoral antibody mediated immune response, we therefore need to understand that this T helper cell is actually going to be producing some chemicals called interleukins. So you will also get the mark if you say that they are cytokines. So different exam boards will ask you to say it's cytokines or whether it's interleukins. So with the OCR, they will accept both, but we tend to talk about interleukins. And the interleukin is a chemical messenger. So you've got this chemical messenger that the T helper cell is going to make. Now that chemical messenger is going to be going, and it is going to signal when it binds to the correct B lymphocyte for it to go through mitotic division and then differentiate. So if we're talking about a plasma cell, we've already had differentiation occur, and a lot of students jump to the plasma cell is that the B cell has been differentiated. That's right, but you've got to remember you had to divide by mitosis. So the DNA replication has to happen, the S phase. You then have to have that checkpoint going on, double checking all the DNA is correct. Why is that important? And the reason it's important is because we need to make this lots and lots of copies of the cell, and those copies need to have the genetic information because that genetic information codes for the production of the antibody, and the antibody is a quaternary protein. So we need to understand that the cell goes through mitosis and then it will differentiate into the plasma cells and the memory cells. So don't forget about mitosis at this point here. That's really important that you're remembering it's mitosis that you have. And guys, if you're just joining me and you are want to do revision, Please do check out where I am as well, because I'm showing the screen and I'll be writing, and you'll get to see some notes, and I'll also answer questions that are there too. So please do pop over if it's helpful and you're a visual learner. If you just find it useful listening to me and you like the sound of my voice, then thank you. Keep staying here and liking and sharing. But anyway, back to this. So we've got our B lymphocyte, which is going to go through mitotic division and then differentiate into either the plasma cell or the memory cell. And it's the plasma cell that's the only one that's going to actually produce the antibodies. Then what we need to understand is our T cells, if they're not T helper cells that produce the interleukins that activate the B lymphocytes, they may be T lymphocytes which are killer cells. Now, if they're killer cells, what they're going to do is they're going to produce, for example, perforin, or you might want to say that it is hydrogen peroxide that they're producing. If they're going to ask you to name a chemical, you want a chemical that's going to disrupt the cell. So these T killer cells, or because you said, Simon, I think it was Simon or someone else, Carpe Diem actually, you said you were AQA, you call them cytotoxic cells, killer cells. But just is that kind of specificity that you need to know for your exam board, because you want to be using the terms that they've asked you in your specification. So our T killer cells are going to produce the perforins, and they're going to therefore be able to break apart the cell that, for example, might have the virus hidden inside. You also will have some T memory cells. So remember, to make more cells, we have to have mitosis occur, and that mitosis is going to be then followed on by differentiation. And differentiation, remember, when we differentiate our cells, we are switching off genes that are not required. So when cells differentiate, they differentiate in lots of different ways. They differentiate in the types of proteins, for example, they have on their membrane. They differentiate by the shape and how much of the membrane they are. So if they are, for example, a root hair cell, they've got a large extension. If they are a neuron, you've got that long axon extension. So that differentiation process is about ensuring that the cell is able to do the job that it is required for. So in this case, the T helper cell, it's needed to help activate other cells, and it activates those other cells by producing these interleukins. So if we were looking at a question that might, for example, ask you to, let's say, describe the role of T lymphocytes in the development of immunity from, I don't know, a virus like polio, then what we're talking about, the first keyword we want to be mentioning is the use of clonal selection. So we need clonal selection to occur. We need clonal selection because we need the selection of the right cell that's required for the attack against this virus. Well, what do we know about this virus? This virus is going to be hidden inside the host cells, and it's going to be using the machinery inside the host cell to make more copies of itself. So if it's inside the host cell, it is therefore going to be releasing either its DNA or its RNA, and then it is going to be using the rough endoplasmic reticulum, is going to be using the Golgi apparatus, it's going to be producing lots of copies of itself, which it will then burst out of the cell, making more copies of itself. So it's like a vicious cycle that goes on. But it is important to know that you have to understand that it's a clonal selection. We're cloning, we're making genetic copies. So clones are genetically identical cells, and because they're genetically identical cells, that therefore means that they have all the genetic information that is required to do the job that they're going to do. So what have we got? What do we need to think about? Well, before we can actually really fully understand how the cells are going to work, I'm going to take you on a little journey. So what do we have? Well, B cells have unique receptors, and they are specific to the antigens. So here you can see I have my plasma membrane, and that has got a receptor on it. So the receptor is where you are going to have this binding of the antigen that's able to bind to the receptor. Now, for those of you who have seen this before, you will know that that is the shape of an antibody. So what the B cell does is the B cell has actually got a receptor on the surface of its cell which has the antibody, which has the antigen binding site. So we know that an antibody has an antigen binding site, and therefore we also know that the antibody has got a receptor binding site. So the B cells have these receptors on the surface of the cell, which allows them to actually encounter and bind to any of the antigens that might be circulating in the body. So when an antigen is going to bind to that receptor, the B cell will process the antigen. And what will actually happen is the B cell will present the antigen on the surface of its cell. Sorry, give me a second. I just need to move some of these diagram labels across. So there I have my B lymphocyte, and I have my receptor, and my receptor has bound with an antigen. So what's happened now is my B cell has actually processed that antigen and presented it onto the surface of its cell. Then what it's going to do is a T helper cell is going to recognize the polypeptide on the antigen presenting cell. Well, the antigen presenting cell might be a B cell that encountered it itself, or it may be a macrophage. So what's happened is we've got a macrophage that does phagocytosis, and when that macrophage does phagocytosis, it is going to engulf and digest the pathogen. It is going to produce a phagosome, then its lysosomes are going to move and fuse with the phagosome, therefore creating a phagolysosome, and then that is going to result in this digestion because of the hydrolytic enzymes that are present in the lysosome. The lysozymes, they will therefore digest down the pathogen, and then the macrophage will then be able to present it onto the surface of its cell. So when we're talking about an antigen presenting cell, that antigen presenting cell has presented the antigen, this polypeptide, on the surface. Now, your teacher may have called it the MHC complex, they may not. Don't worry about that. That is just the place, a very specific complex, which the cells have to be recognized of being, oh yes, we've encountered an antigen or not. We don't want the immune response to be happening too quickly. We don't want the immune response to be happening when there is no need to do it, because that would actually cause damage to our cells. So we want it to be very controlled when it does happen. Tilds, hi, how are you? I see all your lights coming through. Sorry, I was in a bit of a flow there. So remember, and I've done this because I want to identify with you, you can say interleukins, but you can also say cytokines. So do remember that both are acceptable. So interleukin, so the T helper cell is going to produce interleukins or the T helper cell is going to release the cytokines. Either or, it's still okay. Teals, if you haven't done already, do check out my 3.2.1, I think it is, transporting plants videos, because there is specific ones on translocation for you. Okay, so what do we have? So the B cell has presented the antigen. It has got the release of cytokines from the T helper cell. Now it's going to become fully activated. And because it becomes fully activated, it will now rapidly divide by mitosis. And because it's rapidly dividing by mitosis, that is what we call clonal expansion. So I spoke earlier about the clonal expansion, so I'll bring this back down. She says. So we've got that clonal expansion is happening. That clonal expansion happens with both of them. So clonal expansion with the B cells, because they have to go through mitosis, clonal expansion for the T cells as well. If we are clonal selection, we are making sure we are genetically identical cells being produced. Those genetically identical cells have got the correct antibodies that are needed. So clonal selection is ensuring that the correct plasma cells that make the correct antibodies are the ones that are going to be selected for. Do you need to learn gram-positive and gram-negative? Depends on the exam board. OCR, no. You don't need to know it for that one. But you do need to know it for AQA, and you do need to know it for EdXLB. Okay, so what do interleukins do? Well, interleukins obviously stimulate the B cells to go through the mitotic division, so clonal expansion, if we've selected the right one that produces the right antibody. And the only way we can do that is, going back to this point here, is if the T helper cell that's releasing the cytokines is going to fully activate the one that has the correct antibody. So we need to ensure that it is the correct antibody, the cell that's going to make that antibody, that is selected for, and then the expansion is that process of rapid division by mitosis. So the T helper cell releases the cytokines that is going to activate that, but the T helper cell releasing the cytokines also actually signals for our phagocytes to come to the area so that they will do phagocytosis. So that's another thing to be sure that we're not just thinking about it only to do with our B cells, our plasma cells, our antibodies. The T helper cell is also going to be saying, hey, we've got a problem. Come over here. Quick, I need you to engulf and digest this foreign polypeptide that I have recognized as not being a self cell. The fact that it's an antigen means that it is a non-self antigen, not a self antigen, and therefore we need to fight against it. Okay, so we have the interleukins that are stimulating the phagocytosis. We've got the interleukins which are stimulating clonal expansion. Well, the T killer cells, as we said before, what they're going to do is they are going to kill the cells. That's where they get the name from, the T killer cells, because the hydrogen peroxide and the perforin are going to destroy the membrane. So they are destroying the membrane of the cells, but they are not specific in what they're going for. They are basically going to they're going to destroy and kill the infected cells, but they also do the ones surrounding it, which is why, like, you'll get this feeling of, oh, my throat's a bit scratchy. And yet I feel like I'm coming down with something. I can feel the scratchiness, but it's not actually become anything too severe yet. And that's because your body has to actually destroy your cells, especially if you have a viral infection, because that virus is hiding inside your cells. And so you have to destroy your cells for them to then be repaired. So you need mitosis to occur to repair the cells which have been destroyed by the T killer cells. And that's where your multipotent stem cells would come into a question, also linking potentially to the immune system. So it's about always thinking through, right? Well, what else and where else could they be asking these questions? And a lot of the time, a lot of you, when you're doing your revision, you get so honed in on, oh, I must memorize the immune system. And you only learn that part, but you forget, well, what's a receptor? Well, that's the biological membranes topic area. The fact that I have a protein with an antibody sticking out of it, or is it a glycoprotein? Is it a glycolipid? What is it that I have got which is my receptor? In this case, we have the antibody attached to a receptor. So it's really important that you are thinking about all the different topic areas and how they interlink. And then obviously you've got the fact that you want to talk about the differentiation that's occurring and not just linking it to the process of mitosis would be my last little advice for you. Okay, so let's just have a look at antibodies then and antibody production. Let's go there. 1 second. Okay, so what is an antibody and what is its structure like? So you should know about the antigen binding site, which is specific to the antigens. But a lot of the time, again, students memorize these words. They memorize that this is what's happening, but then they don't go a step further and think about, well, what is an antibody? Where was it produced? How was it produced? What could they link that to? So antibodies have got different things in which they are going to do. They could be antitoxins. They could be agglutinins because they are going to do agglutination where they cause the pathogens to stick together. Or they could be opsonins because they're going to do opsonization. Now, if you are AQA, because some of you I know are AQA, you need to know about neutralization, you need to know about agglutination, and you need to know about phagocytosis, but you don't need to know the words opsonins, agglutinins. However, OCR do. You need to know those words because they will throw them in there for you. So an agglutinin is a type of antibody that will cause the pathogens to stick together. An opsonin is a type of antibody that is going to make a pathogen more recognizable to the phagocyte. An antitoxin is going to bind to the toxins. Because it's binding to them, it's neutralizing, they're not able to cause a problem. So make sure you are revising your keywords which are in your specification. So you should be using your specification alongside when you're doing your revision. And I say that because I always, when I'm doing my tuition sessions, I always have my specifications open. I don't have it open right now because I am doing this live for you. But what I am going to do is I'm going to open the two of them and show you what I mean. So here we go. So if you are OCR, which is the one that I have built my resources for, then you want to look at the parts of your specification that show you this. So this is in communicable diseases, and this is looking at the Structure and modes of action of the phagocytes, specifically the role of, as it says in the specification, opsonins, and then with regards to moving on through the specification, an outline of the actions of opsonins, agglutinins, and antitoxins. Now, if we are looking at AQA, because I've just opened yours up, didn't, let's try that again. With yours, you need to know. Second. Actually, let me just bring this in. Parts of your specification that it's important that you are able to use. I'll bring that across here. Is, as it says on the specification, that you are looking at the antigen antibody complex. You need to use those words. The destruction of the antigen leading to agglutination and phagocytosis of bacterial cells. So that's specifically showing you what it is that you need to know. So I would always advise, as you're doing your revision, having your specification open and double checking that you have got those keywords, because if you're not using those keywords, you will potentially watch a video like my video and hear me say something that, like opsonins, which is specific to OCR. It's not wrong what you're saying, and therefore you should still be credited the marks. However, it might get missed by an examiner, and that's not intentional. That could be a mistake. It could be that they were going through and it wasn't part of the additional information that was given to them. They hadn't done it because it's not part of your specification if you, for example, were AQA and use opsonins. And so what I would say to you is be aware of that and be aware of the key terms for your specification, and make sure you have an up-to-date specification. Also, make sure you're using a textbook, if you're using it, or a revision guide that is specific to your exam for the period when you are sitting it. Okay, moving on. So I want to talk about these antibodies, and I want to go back and look at the structure of the antibodies and how they're actually produced. So we have got our B lymphocytes. Our B lymphocytes that we know are going to go through mitosis and differentiation. We know that the plasma cell is going to produce the antibodies, but how does it produce the antibodies? Well, this is where you bring in your nucleic acid nucleotide knowledge, which is about protein synthesis. You know that your DNA, your chromosomes, now we're talking about eukaryotic organisms, so I'm just going to start bringing in some other knowledge where, again, a lot of you don't think about these things, and maybe it's just how my brain works, I don't know, but if I'm being asked about protein synthesis and I know I'm looking at a B cell, the first thing I'm going to do is I'm going to say, right, this is eukaryotic, and it's eukaryotic because it has a nucleus. And therefore, because it's eukaryotic, I also know that its DNA is linear, and I also know that the DNA is associated with histone proteins. Oh, crumbs. Cannot write to save myself, and my writing is atrocious. I know that, guys. I do apologize. Histone proteins. The funny thing is, I'm writing on my board here, but I used to write so much in my lessons as well on the actual physical board, and I would be up writing all over the place. Then I'd be rubbing it back out again. Then I would be rewriting it. Whereas this way, I get to keep all my notes. The number of slides that I have with all the notes and how I teach things, it's, yeah. Anyway, little bit of divergence there. Is Edexcel a similar exam board? Do you know what? I don't actually know about Edexcel A. I've taught Edexcel B. I would assume that Edexcel A would be very similar, and it would be very modular on the way that they teach it, because Edexcel B is more holistic, but I wouldn't know. I just watched a video. I just had a quick question. The co-transport of hydrogen sucrose. What's your question, Tilde? Sorry, I've just seen your question there. Do I'm trying to have a quick look. Nobody's writing anything on my other thing. Actually, it's probably just me. Is this also about Hi? Is it also about cytochrome C? Cytochrome C is a specific protein which we tend to talk about when we're looking at respiration. But if we're talking about cytochrome C, it is a protein that had to come through protein synthesis. So we're looking at, until, thank you so much for all the likes, by the way. I really do appreciate it. From where to where does the co-transport occur? And then how does it affect the water pressure? Be careful on your words of water pressure, water potential. So first of all, the co-transport, and I'll come back to this. I don't even know if the person who was wanting me to do immune system is still here. So Tills, because you've asked the question, I will jump to that. You have said, where to where does the co-transport occur? So we're talking about from the apoplast, from the cell wall and into cellular space. You want it to go into the companion cell. It's going into the companion cell because the sucrose, basically the sucrose gets co-transported out of the source cell. It can then flow really fast and without any hindrance between all the cells through the cell wall and into cellular space, and then gets brought back into the companion cell to then be moved into the phloem. And when it gets loaded into the phloem, that's when you're going to affect the water potential. Because you're affecting the water potential, that's going to move water by osmosis in, because you have lowered the water potential. And because of that, that's then going to create a high hydrostatic pressure, and that's going to force the water and the sucrose and assimilates amino acids in both directions to wherever there is a lower water potential. Anytime I have a bio end of, does it really matter? Sid, yes and no. I'll come back and answer that question. Is hydrolytic pressure similar to water? No, hydrostatic pressure is different to the water potential. Hydrostatic pressure is about the fact that water is incompressible. You have the phloem cell walls, which have cellulose that make it really strong. And so therefore, because if you fill up a bottle of water, so here I have a bottle of water, bottle of squash, but I have a bottle. Now I don't have it completely filled with the water, but if this was a plastic bottle, like a proper plastic bottle that I could really squeeze, you would be able to squeeze the top. I can squeeze it actually a little bit at the top because there's air and there's space between the air molecules. And so therefore I can compress that down because it's a gas. Now a liquid, because they fill the container, if it is full with a liquid and I try to push that plastic bottle again, it will not work. That's because water is incompressible. We cannot force it together. So because we can't force it together, it creates, as it bounces off the phloem wall, it creates a hydrostatic pressure, and that hydrostatic pressure is therefore going to cause water to flow to wherever there's less of a hydrostatic pressure. So it flows from a high hydrostatic pressure to a lower hydrostatic pressure, which is away from where it was loaded. Sid, I'm coming back to your question. You said your triple science in year 10, you have a bio end of unit test. Does it really matter? Yes, it matters because it will identify what you don't know now. And so you can start filling those gaps. It also shows you how to improve by making the mistakes in a test, end of unit test, end of topic test, end of mock, end of year, whatever test that is that they're giving to you. It's going to identify what you don't know and where you need to focus on. So yes, it does matter. Does it matter in the grand scheme of things? Well, it depends. Is it I haven't done my revision and I'm not going to be able to revise before the test? Therefore, you're finding out your baseline level without any revision. Next time, make sure you revise, see if you improve. And then it shouldn't matter as long as you do put in the revision at the end. But what tends to happen is most students don't do the revision until the last, just before the exams, they cram it. And when they're cramming, that's then going to result in their brain going, it's too much, I can't do it all. And so you can't retain all that information. But if you're slowly doing it over a longer period of time. It's called space learning. What's going to happen is you're going to end up where, because you've done it again and again and again and again, it just gets ingrained in your brain. It's boring, it's annoying, it's frustrating listening to your teachers. Like if you watch any of my live sessions, I'll repeat myself again and again and again. The reason I do that is because the more repetition you do, the more you're going to get that commitment to long-term memory. I know that my working memory is shocking because of the fact that I have ADHD. And so therefore I have to find ways to make it work. I have to find ways to make it stick. And so that's where it matters. So the answer is yes and no. But if you're already in year 10 and you're not preparing yourself for these tests, then what I would say to you is you're setting yourself up for a really hard couple of years, because it gets more and more complicated. Right now you're GCSE, and so therefore that's a nice place to be compared to my A-level students who are on here right now. However, the more foundational skills you have, the better. I am doing lives for free on a Monday at 6:30. I've done 2 lives. Go check out on my channel if you're interested in maybe a bit of a recap. There's also some videos, a little bit on chemistry and a little bit of biology, that might help. Right. Here we go. Tills, I'm coming back to you. I'm just double checking if there's any other questions on that. Where are you? This is kind of related to transporting plants, but as AQA, do I need to know about the cambium? Let me double check. I feel like you do, but off the top of my head, I couldn't tell you. That's the thing about the different exam boards. Different exam boards have different parts that they have really honed in on that you need to know. I'm not sure you do, but again, let me just double check. Phloem, xylem. It'd be in the stem cell part. If it is, let me just... If anyone is on here, either past or current AQA, who could answer that question quicker than me, then please do. Right, here we go. Stem cells, pluripotent, totipotent, induced pluripotent, unipotent. I don't think you do. I don't think you do. I'm in year 13. Do you know, sir, any tips on exam technique on application questions, as they're really tricky? Frosty, what I would say for you on that one is, yeah, they are really tricky, but it's about, one, thinking outside the box. So many students will look at a question and see that this question is on, I don't know, muscles. Let's go down the route of muscles. So the question will be on muscles, and they'll be like, yep, right, okay, I know my muscle content. And then they will actually not be asking about muscles. They're actually asking you about proteins, because they're asking you about the, let's say, the troponin. And so you get hung up on the muscle side of it, the content for muscles. For example, the calcium ions are going to bind to the troponin is going to change shape, blah, 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 all that stuff. And actually what they want to know is, do you know that the troponin is a protein that was produced through protein synthesis? And therefore, transcription, translation is what they're actually assessing you on. And that's what I would say is where most students lose the marks in an exam, because they don't look outside of the question and think, what is this asking? They don't have the words. What I mean by this, I don't have the words, but they don't have the words that come to mind. So if I were doing a question, whenever I'm doing, like tonight, I was doing a question with student in my session, and I said to them, okay, what are your key words when it comes to, for example, a question that says anything about an enzyme? Now there should be like, it should come straight at you. This, and this. Like when I did the eukaryotic cell, I went straight away. Eukaryotic, nucleus, DNA, which is linear, associated with the histone proteins, membrane bound organelles. I should add that in. So these are like my go-to answers. They're my go-to, and I'm like, check, 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 check. Do they relate to the question? Okay, if it's about enzyme, what is it that I am going to be doing? Right, it's an enzyme. An enzyme is a protein. It's a protein that's a globular protein. A globular protein which has got a tertiary structure. That tertiary structure was formed because the interaction between the R groups. What does that mean? Right, well, the interaction of the R groups are what make it have its specific shape. So when the active site and the substrate come close together, if they have a successful collision, they're going to form a slight temporary bond. Now that's going to change the tertiary structure, therefore that is going to be the induced fit model. So it's about having that connection. So going back to the enzymes, and I've just saw a question, do you do private tuition? Yes, it's the only way I can do these free lives for you is through my private tuition sessions that I do. So yes, I do. Oh, that's so cool. Second year of dental student. Wow, that's awesome. That's so cool. Right, hold on. What, I missed it. Oh, Tills, that's awesome about your practical. Which one did you do with osmosis? I had a student tonight, an AQA student, and they have done the osmosis practical, but they did it on temperature, interestingly, and now they're going to do the serodilution. So we were doing a session on serodilutions and how you would use that to make a calibration curve, and then how you would be able to do that to work out the water potential of the sample of whatever it is that you're looking at. So which one did you do? Oh, and the serodilutions, that's awesome. Oh, Faith, it's so good. Actually, do you know what, Faith, you can't be 2 years ago. Seriously, are you actually? You're in your second year. That's crazy. Because that would have meant, Faith, correct me if I'm wrong, are you saying that you actually were in my sessions when I was just on TikTok before I'd gone to my YouTube channel? Because I started on here in 2023 was when I started on TikTok, because one of my students was like, Miss, have you ever thought about going on TikTok? I was like, TikTok, what is that? So I've had to learn very quickly about TikTok. And then, as I was saying in my previous video, that I, please, for my write-up, could you please help explain what a calibration curve is? Oh, okay. GCSE biology, but I have a mock tomorrow, and I was wondering if I could ask you something. Yes, of course, ask away. And how to calculate what potential for the write-up. Okay, so Tildes, whilst AK47 is writing the answer, his question, sorry, thank you. Would you say past questions are the best way of revising bio once content is revised? Yes and no. Past papers are amazing. Don't memorize mark schemes. Make sure that you are assessing yourself and improving your answers. The problem a lot of students get is they do just memorize mark schemes, and that doesn't help. So past papers along with content, figure out what you don't know, go back over the content of the stuff that you don't know. So use the past papers as a way of identifying your knowledge gaps and fill those knowledge gaps. So whenever I do my sessions with any of my students, I'm always asking them questions. They hate it. And I say to them, you need to be comfortable being uncomfortable, because if you can't answer me the questions that I'm asking you, you don't know the content. So I need to fill those gaps. So you need to figure out what you don't know, use that to go and do your revision, and then come back and try it again. Also, do it again. Even if you've done a past paper question, do it again, but this time check your answer from previous and see if there's any bits that are still not sticking in your head. And another alternative is to do a paper. If you can't answer a question, use the mark scheme to write the perfect answer, and then do the paper again and see if you were able to regurgitate your own perfect answer in your own words, not just rewriting the mark scheme, because that's a waste of time. You need to be comfortable. Yeah, absolutely. Be comfortable being uncomfortable. I'm always uncomfortable. I'm like, I've got 40 odd years on me that I have that experience. If you'd have told me your age it was to be comfortable being uncomfortable, I would never have said that that was possible. I was always uncomfortable. Anyway, back to your question, Tills. So a calibration curve, you did a zero dilution. Was it sucrose, glucose, or salt that you used? What did you actually use for your, it was glucose. Sucrose, okay. So you did a zero dilution where you took a sucrose stock solution and you actually diluted it down to get you different concentrations. So 0.1, 0.2, maybe 0.4, 0.6, 0.81, something like that, depending on what your school actually did. So you had known concentrations because you did the zero dilution to control the outside environment. Then you put your plant cells, so you would have used a cork borer or used, like, just cut up the squares. So you will have cut it up and you would have put it into this known solution. You weighed it before, you weighed it after, so you were looking at the mass and the mass change, and you would have worked out your percentage change. What you would have then done is graphed that out. So by graphing out your potato chips and whether they increased in mass, whether they decreased in mass, or whether they stayed the same, that would have allowed you to plot that against your 0.4, 0.8 concentration. And that therefore means that if they increase in mass, because on your y-axis you should have 0, it kind of looks like this, so 0 and positives and negatives. So if it increased in mass, you would plot them above. If it decreased in mass, you plot them below. And so therefore when you draw your line goes through wherever it crosses at this point here, that's your isotonic point. So therefore, by using known concentrations outside, you can figure out what the concentration was inside the cells. So you're using the known concentrations to find the osmotic point, i.e. the isotonic solution, and that therefore means for every one that goes out, one will come in again. And so that means you have the same concentration of stuff, solutes, on the outside as you would have had in the inside. And then you can do that as a comparison against like sweet potato or beetroot or normal potato or whatever it is. So when isotonic point is 0, it means water potential of the tissue. Yes, that's exactly what it means. Guys, thank you so much for the likes and the shares. If I'm not answering your questions, I do apologize. I'm not trying to miss it. I'm trying to keep up. This is obviously being recorded elsewhere, and I'm chatting to you rather than over here. Nobody's talking to me on YouTube, but never mind. Is it okay to do questions from other exam boards? Frosty, absolutely, yes it is. However, here is my little warning to you. I always use other exam boards questions because I think it is a good way of helping you see other questions. However, I know the spec for OCR A inside out. Yes, I still double check against the specification, but I know it. I know the course. I know I built my channel around that course, so I know it inside out. If you are doing questions from other exam boards, just be careful you don't start learning content that you don't need to know. So for example, AQA, they do digestion, you don't in OCR. They don't do topic areas like let me think this one through. What don't they do? The brain? No, it's not the brain. Is it the brain? There's a big topic area they don't do, but I can't think right now because I'm on my 4th hour of teaching back to back. So yeah, just double check against your specification would be my only little worry. I would have liked to have studied biology when I was in secondary school. Oh, Nina, what did you study? I only studied biology. I didn't do chemistry or physics, and then I had to teach it as part of my secondary qualification, and I did it for a long time. Actually, to be fair, every time I go in, I always get given the classes that are struggling, and they're always like, wow, you teach in such a different way. So I actually do really like both sides. Immune response and respiration I want to do. Oh, that's so cool. The key thing is, Frosty, what I would say to you, if you love those topic areas, don't study them. Not at university. I'm not talking about university. I mean right now. Don't study them now. Use the fact that you like them, that you feel confident with them. They're like the last of the things for you to study. Right now, those topic areas that you don't want to do, those topic areas that you're like, oh, I absolutely hate that, that's where you need to go. That's what you need to study. And the reason being is because you don't want to avoid it because you don't like it. I would say definitely a big learning thing was doing the hard stuff first. I hated genetics and now I love it, but I still hate it sometimes because I'm like, ah. How do to make flashcards for biology? Okay, so how do you make flashcards for biology? How would I make flashcards for biology? How I would make them is I would start with, I'm just going to throw something out there, non-cyclic photophosphorylation. So if you are doing photosynthesis right now, you're doing the light dependent reaction, you have to know about non-cyclic photophosphorylation. So I would have, for example, either, I'm very visual, so I would have a picture of it in front of me, or I'd have the word. So it depends what you want to do. Will this live be available to? Yes, Maria. That's why I do it on here. Sorry, I've got someone answering me a question on my YouTube channel. Yeah, I do them on here so they're always available for students to watch back. Because actually I want it to be that there might be something that's good and useful for you, even though I obviously do chat and answer questions. As we get closer to the exams, I will run more live sessions as well. But flashcards, I would do photophosphorylation, non-cyclic photophosphorylation one side, and then on the other side, I'd only write 4, maybe 5 bullet points maximum, and they would be short bullet points. I wouldn't be, I've had it where in the past students have written like so much, like the whole course in the tiniest of writings to fit it on one card. That's not what it's about. What you want to have is like your flashcards or your cheat sheets. Your cheat sheet that you go, oh, it's a question on non-cyclic photophosphorylation. What do I need to talk about? Right, what am I talking about? I'm talking about photosystem 2, photosystem 1. I'm talking about proton pump. I'm talking about reduced NADP. I'm talking about ATP synthase. I'm talking about photolysis. And I'm going to talk about water and light. They're my, okay, I said 5 and I've just given you 8. But they would be my cheat sheet bullet points. They're the bits I want to bring in when I talk about non-cyclic photophosphorylation. So that would be my advice on that one. Is biomed a good degree to go into? I don't know because I never did it. I think it's a very good degree to do, but I don't know from personal experience. But I would say, yeah, it's a really good one. I have some of my students who are doing biomedicine in lots of different universities and they're absolutely loving it. So yeah, absolutely, I would say it's a good one. But I don't have personal experience on that one. Dihybrid Punnett grids. Honestly, I almost failed my whole 3rd year of university because of genetics, but I passed on that 4th attempt and then I got 2-1. So it was amazing. Do you know any websites, YouTube accounts who tutor the new? No, I don't, Elena. I'm really sorry on that one. I don't. It was on my list of things to potentially do, and I have had a few teachers who have asked me to build some resources for them, but at the moment I haven't had the time to look at it. Which one are you doing? The applied science, just the applied science one? Or are you doing the human one? And there's a 3rd one, which is the technicians one. I think it's the technicians one I was asked to do the stuff one. A lot of the content is very much overlapped though. How did your top students revise? What were their... Depends. Different people have different ways in which they revise. So I had a top student. I'll give you an example of a top student I had. So I had a top student and I picked them up in September of the year 13. Now they were a top student getting A's, okay? They were getting A's, B's and A's in the assessments that I was setting them when I first started teaching them. Now they'd read the textbook 3 times over and they knew more of that textbook than I did, but they didn't have the application skills. And so they thought reading the textbook was their way to go because they were memorized, they were learning it, they found it really easy, and then they would get these questions and they couldn't apply the content to the knowledge that they'd gained to the questions. So I had to teach them a new way of learning. I had to teach them a new way of thinking about the world around you, about biology, how it is it's everywhere. You give me a topic area, I'll find something biological to talk about. Because that's how you've got to see the world. That's how the questions want you to be able to apply your knowledge. So it really just depends. I had another student who bought every flashcard that they could, but didn't ever use them in the right way. So it really depends. For me, I know I have to do little, often, and go back over and do the same stuff again and again and again. I forget things all the time. And it's hard. It's hard to find what works for you. And it's not that it's the same for everybody. And so your friend might find one way really useful for them, but that doesn't mean it's useful for you. It doesn't mean theirs is right and yours is wrong. It doesn't mean yours is wrong and theirs is right, or whichever way around I've said it. It just means it's different, and that's okay. But you've got to find what works for you. I found mine in my 4th year of uni. Luckily, I got into my 4th year of uni and everything counted for that year. And I found my way of learning, and that was it. From then on, it kind of progressed. Maria, what did you say? Your channel really, I would also appreciate if you do some exam question walkthroughs on specific topics for OCR A A-level. Maria, I have done a lot of that in previous lives. I have been considering doing the papers, specific papers, and working their way through. But at the moment, it's trying to keep that continuity. I've got lots of things that I'm doing, and my tuition that I do pays for me to be able to do this. So yeah, it depends. I will try and do some more, but there's definitely videos of me doing walkthroughs, especially if you watch any of my lives that are on here, that are saved on my channel. If you watch any of them, you will actually see that there's loads of walkthroughs that I've done. I used to have a student last year, Amna, who was awesome. She would come to my lives, like these lives like I'm doing right now, and she'd literally go, Christine, can you look at 2021, this question, that question, can you go through it? And that's what I would do. I would literally go, yep, sure, pull it up, and we would go through it. So they are there. If you go and watch the lives, you'll see the ones that I've done with lots of different questions. But I will do some more as we go. Science teacher, final boss. Thank you, if that's what you're saying. I am. I love this. The AI is saying it's all about dihybrid Punnett squares. Ask your questions. That's brilliant. Just find this live. We'll check your YouTube. Oh, thank you. Please do, and please share it. I promise you, if you're OCR, the whole course is there. What are the grade boundaries like? My college says that an A star is 90%, but people are saying otherwise. Chemistry, absolutely. That is 90. It was 90% for an A star in chemistry, not for biology. Biology last year was 79%. Hold on, I have the spreadsheet. Oh, it's 10 o'clock. I'm gonna go soon. 1 second. What was it last year? Hold on. Yeah, 69%. I did think it was 60. It wasn't as high. So it was 69% for an A star last year, 59 for the A, 50, well, actually rounding up, for the B. C was 42, 33, 25. So yeah. I really hate how exams are all dependent on keywords or phrases as opposite to actual learning for. It's not, but it is. It's really hard, and I get that because it can feel like it is really tough to know what the examiners are actually asking you. The key thing to know is those mark schemes that you're using to memorize from, they were the mark schemes that were available that year, and they won't be the mark schemes that are available the next year. And the reason being is because the people who are the examiners and the senior examiners, when you have sat your exams, they collect your papers, they start marking your papers, they do a sample, they all get together after they've marked a sample, and then what they'll then do is they'll then agree and disagree what they'll accept, what they won't accept, based on, so there's a mark scheme already there that they mark against, and then they tweak it. And that's why you'll see allow this, do not allow this, allow this, do not allow that, ignore that. And that's because they've seen those answers come through and they've decided that they're not enough, or that they're wrong and they're shouldn't be allowed, or that they will allow it because actually they're on the right line. So that's what's for that year's exam. Now, that doesn't mean it will be absolutely the same, and that's where memorizing mark schemes are not the way to go. They're not going to help you. Okay, it's really important you understand that, because that is not a useful way. How do I prevent myself from blurting out all the knowledge that I know on an exam question? So blurting is a good thing. I would say annotating around the question is a good thing for you to just formulate a response in your head. So I wouldn't blurt it on the answer line. If I was going through the question, I would be reading the question and writing little notes to myself. Now, it may be that these parts of the information that I'm writing are going to come up in the question later on. That's okay. What I'm doing is I'm priming my brain and thinking about what I already know before I get down to that question. Because a lot of the time, if you prime yourself, you will then have got the thinking done and taken the time to read the question before you start answering it. What a lot of students do, and this really frustrates me, is they skim read and then they just start blurting everything and anything. And I can tell they haven't read the question. They haven't fully digested what that question's asking them. Because they haven't fully digested it, it therefore means that they're not giving me an answer that is going to get the grades. The other thing I would also say to you that is hard for a lot of students is students give me GCSE answers all the time. As an A-level student, they give me an answer which is a GCSE level response. They don't go into detail. And because they don't go into detail, they miss out on the marks. So if you're talking about carbon dioxide concentration, for example, and photosynthesis, I would be expecting you to say carbon dioxide concentration that's needed for the Calvin cycle in the light independent reaction. Photolysis that occurs in the light dependent reaction where water is split apart. It's that kind of detail that's missing. So the papers that I reviewed this year, so when students sit the exam, after they've sat the exam, once they've got the results back, for a few students, I review their papers and I have a look at their papers and I see, would I submit them in for a remark? Would I not? Do I think I would give the mark? Would I not? I'm a harsh marker, and I know I'm a harsh marker because of the year's experience that I've got, especially when I'm doing mock papers. But when it comes to the real exam, we don't want to just fail you. We don't want to remove the marks. We want to find those marks. But if you're not making it possible for us, there's nothing we can do. We are doing a process which is standardized. And the reason it's standardized is because it is a recognized qualification from such a vast number of places that people actually come here to the UK to study their A-levels because it's so strict and controlled to allow for it to mean something rather than just I think you're about an A star. I think you're about a A. I think you're a E. I think you're a U. That's not for me to make the judgment. That's the assessment done at that time. Now, that's great if you're good at exams, but if you are like me and not very good under pressure in exam situation, I can't write fast enough for the time that's needed. I stumble and do things back to front. That's what I would say is a problem for a lot of students where, one, they don't go into detail, two, they don't know the content, and three, they assume that examiners know what you know. We don't have a clue. I don't, like right now, I'm talking to right now I can see Frosty's now because he was the last question that I've got in front of me. And so therefore I'm talking to a name that I have no idea who they are. When I'm examining, I have a picture of your script. That's it. And therefore, I am marking a picture of what I can see. I don't know who you are. I don't know what you know. I can't ask you and prod you and do the things that I would do in an interview face to face. So therefore, we have to take what you write as being your answer and your knowledge. And that's why students lose marks. That, and you'd be surprised how many make silly mistakes. They don't double check or proofread their answers. Where it's so easy to drown in jargon. I would say find a way to limit it down to the lowest possible word count, which is really hard. I'm a bit more, I find it a bit easier doing that. I find it harder to, although I talk very much, but my written ability is like, I'm like, I do this, that's it. Whereas I'm like, if I'm verbally doing something, like I can talk for Great Britain, as you can see, because I'm still going. But when it comes to writing things down, I find that I'm much more restrictive, is probably the word I'm looking for, in how and what I write down. So yeah, I would say my advice on that would be learn to summarize. Learn to summarize. Don't get AI to summarize. Learn to summarize, because it's funny what AI is saying, even on this tonight. Excuse me. Oh, Sarah, that's horrible. I'm sorry you're losing your voice as well. Right, guys, I am going to finish. Oh, AI's got it right now. Biology exam tips. Yes, it is. Go watch, please do go watch one, because I get paid. If you watch an advert, I get paid for that. Woohoo! But yeah, please do go watch my videos. I made the whole course free for you. It's all there. The whole 2 years course, and it is specific to the specification points. So please do go watch it. Oh, thank you. Please do comment, ask questions. Please do like, share. So yeah, please do. Can you write smart schemes for questions that are more simple? Yes, Frosty, yes, memorization of things like that. But I would still not memorize it word for word. I would memorize keywords. Keywords are the same regardless. If it's talking about transcription, those keywords are really important. That is a good use of your time. Yes, I would say that. Right, guys, I am going to go because it's 10 minutes past my bedtime. Could you scroll up? I said some stuff. Oh, did you? What did you say? What did you say? What did you say? I missed it. I thought I'd got everyone. I have no idea where you are. There's just so many other people. IP3 is your problem. Oh, I missed this. What did you say? Don't like how my future is based on my exam. That's really hard. I know I could have done way better. I missed a whole load. I'm so sorry, guys. I've been here in ages. I ended up getting a C. I'm in uni right now, but I don't know if I should reset. Ah, right, okay. Why? My question to you: why would you reset? What will you gain from doing the reset? So right now you're in uni. You're already there. What do you want to gain from a reset? Is it that you want to switch universities to do a different course? Is it that you don't like the course that you're doing? Because I would say trying to transfer potentially, seeing if that's an option, if you're already there. If you're going to resit, you're then going to have to pay as a private candidate. That's a lot of money added in. Have you got the time to do that? Are you going to give up the uni course? Like, there's so many factors you need to think about when making that decision. So I don't know. Have you written all the reasons as to why you want to? Right. I don't like the course I do. I'm not happy with the grade. Ignore the grade, okay? Ignore the grade. So and I know that sounds weird me saying that to you right now. Ignore the grade. So you got a C. You're not happy with getting a C. However, I got a B in biology in an AS course. Okay? So I did my hires in Scotland and I got a B in my higher biology. I got a C in my English, I got a C in my maths, I got a C in my PE. I did that. I didn't stay on to year 13 equivalent in Scotland. I didn't do my advanced hires. I left at the end of year 12, which was 5th year at school reasons. But I left school. School wasn't for me. Now, I currently sit here having left school, worked for a year, went to college for a year, didn't carry on to do the second year of college for reasons, took another year out. I went to university when I was 20 years of age, and I then did my degree, and I only really got into my degree by the 3rd year, to be honest. I mean, the first year was just about having fun, making friends and whatnot. But what I would say is I discovered who I was as a learner, who I was and what I was capable of later on in my university course, because I actually got some fantastic lecturers who inspired me and pushed me and saw potential in me. And I had to do some courses that I hated. I had to do things on, sorry if you're a geologist or a geographer, on things like rocks and the different rock cycle things. Like it was rough, but I had to do it. I sit here right now with not only a BSc Honours, but I then went to Cambridge, and I've been to Cambridge and got 3 different qualifications from Cambridge. So I have my PGCE in science, I've got my PSPD in restorative approaches to conflict and skills, and I also have my masters all from Cambridge. And I'm not saying that to show off. What I'm saying is, if you'd have asked me when I was your age back then, I wasn't going to school, I wasn't going to uni, I wasn't going to do any of that, because I didn't believe in myself and because I hadn't found my way. You have to take all the different factors into consideration. I'm not saying don't resit. I'm not saying do resit. What I'm saying is make sure you've looked at all the possible scenarios before you make that decision. Because I know there's some days I don't even want to do this. I love doing this, don't get me wrong. And I'm like, it's way past my bedtime. I'm obviously having a good night tonight because I'm going a lot longer. But I love doing this. But there are days when I'm just like, do I have to? And you have to understand that it's okay to have those days. It's okay to be not enjoying it right now, the module that you're doing, but there might be a better module coming up. So I would say right now, if you're in your first year of uni, you've only given it 2 months, maybe 3 if you started in September, but most unis don't start till beginning of October. That first freshers period is never really learning. So it's only been 2 months. That's not long enough for you to actually get an experience of what it's like at uni, and it could turn around. Now, if by January you're still feeling the same way, that's very different. But I would say give it a little bit longer, but also look at all your different options. Right, I definitely need to go. Oh. Well, if you're already able to transfer, just spend the year going through and doing it and enjoying uni and being a part of the experience is what I say. Right, I have to go. I have to go because otherwise I won't go. Guys, thank you for joining me. I'll be back on Wednesday again next week. If I've got time, I will do Monday as well. It depends how tired I am because Monday's not my usual plan. But if I can, I will go live on Monday. Thank you for liking. Thank you for sharing. Thank you for watching. And I look forward